Hello, everyone. Welcome to our webinar today. So we are just waiting for people to come in to Zoom, and then we'll start. Wonderful. Hi. All right. Okay, let's start. Hello, everyone. Welcome to another MaxWell webinar. This is our 5th webinar for 2021, and we are super excited to have a special MaxTwo demo today. And yes, so of course, I will show you later what we have. But first, while we are waiting for more people coming in to Zoom, I just wanted to thank you for coming here, for joining us. And I would also like to thank you for having this time this afternoon to join us from here in Zurich. So we are broadcasting live from our biolab. Later on, you'll see our biolab in two views. One is the lab itself, and one is focusing on Giulio. So you'll see that. And you'll also get to see our MaxTwo system in action. All right, so now let me begin. All right, as I mentioned for today, our session is about our MaxTwo demo, which is a powerful system to characterize the function of human iPSC-derived neurons. So for today, we have in our demo live cells that we have cultured. So these are Fujifilm cellular dynamic iPSC-derived glutamatergic neurons co-cultured with astrocytes. So you'll see that quite soon. And my name, by the way, is Obien. I will be your host for today's webinar. And first off, I would like to introduce to you our company, MaxWell Biosystems. So MaxWell Biosystems is founded back in 2016, and our mission is to provide electrophysiology systems and solutions to advance scientific discovery and to accelerate the development of novel drugs for brain diseases. Of course, it can also be used for other electrogenic cells, but you're focusing on neurons. Our core expertise includes the hardware and software development for microelectrode array platforms and also the applications of such microelectrode arrays. So we have already more than 80 journal papers that have been published related to this technology. And our customers are from academic labs, contract research organizations, biotech, and pharmaceutical companies. And some of you in our audience today are also our customers. So welcome. Thank you so much for joining us. All right. So as you can see here, we have our founding year in 2016, and actually this month in September, we're celebrating our 5th year anniversary, and so we are super excited to have this webinar together with you. Just last Friday, we had our team event. So as you can see, we had our cycling all together. We went to some restaurants and did some Swiss sports. And related to our 5-year anniversary, we actually have some special offers if you will purchase a system within 2021. So please stay tuned for more information or contact us directly via email or by Calendly. All right, so now I will introduce our speaker for today. He is our product manager and application engineer, Giulio Zorzi. Hello, Giulio.
Hi, Marie. It's great to be here.
Yes. So Giulio is live from our biolab, and yes, so he will show you how the MaxTwo works and also more about our system. So it's yours now.
Great. So yes, hello everyone, and thank you so much for being here with us today. So I will start with a rapid presentation of the technology, just to have everyone on the same page. And then I'll jump right into the live demo of MaxTwo that you can see here on my side. So I already loaded a well plate, but later on I'll also show you how to do that. So let's start from my slides first. Okay, great. So again, just to begin and to have everyone on the same side, today we're going to, of course, see microelectrode arrays or multi-electrode arrays. But first, I just wanted to have one slide to, let's say, present the two gold standard, I would say, for electrophysiology, or let's say for the electrophysiological characterization of your cells. So what you're seeing here at the top of the slide is one of these two techniques, patch clamp, which basically consists in actually patching your cells with a glass pipette, with a glass patch. And this technique gives you, it has a huge advantage of giving you the full dynamics of the membrane potential, but has also some drawbacks. So first of all, it's a quite challenging techniques that requires a lot of expertise to be carried out properly. Has a, let's say, relatively low throughput. So of course it requires a lot of time, or let's say some time, to patch multiple cells in your culture, and it's usually an endpoint experiment for sure, let's say, for your cells. 99% of the times after you have patched your cell, it will die. Conversely, what you're seeing at the bottom of the slide is what we call micro or multi-electrode arrays. So in this case, you're losing part of the information, so you do not have access to the full dynamics of the membrane potential, but let's say you have some advantages that can outweigh this loss. So first of all, it's a very easy technique to use, so you don't require extensive expertise to carry out your experiments. It allows you to have a much higher throughput in terms of cells that are being recorded. And let's say, maybe not most importantly, but quite importantly, speaking for us, it allows for developmental studies of your cultures. So what is the difference between, let's say, standard MEAs and what we're going to see today? So what you're seeing here on the left is, let's say, an example of a standard low resolution MEA. So these are very, let's say, it's a very simple technology. You have electrodes being routed out to amplifiers. And let's say the first obvious disadvantage is that when you do not have cells lying on top of the electrodes, so the cells that lie in between the electrodes cannot really be recorded. Another maybe less obvious disadvantage is that when you have a situation like this one, in which you have multiple cells on top of the same electrode, it's extremely hard, if not impossible, to reassign the signal to its source. So what we are proposing as a solution, and what you're seeing here on the right, is based on active MEAs. So MEAs that are based on active circuitry embedded in the chip, based on CMOS technology. So this technology allows us to pack a lot of electrodes within a very small area, and be in a situation in which we always have more than one electrode addressing a single cell. So diving right into the technology itself. So today we are going to focus on MaxTwo. What you're seeing here on the left, of course, but also what you're seeing on the right of the slide, which is a self-standing incubator. Let's say MaxTwo in its well plates features exactly the same technology with which we launched into the market back in 2016, so MaxOne. Let's say, the core of the technology, so what you have in the single well, in the 6 well plates, and in the 24 well plates, is a CMOS-based MEA featuring 26,400 electrodes in a 2x4 millimeter square area. Each electrode is roughly 10 micrometers square in size, and the pitch, so the center to center distance in between two electrodes, is just 17.5 micrometers. So this really allows us to have always more than one electrode, as I said, addressing the soma of each single cell. On top of that, and I would say this is together with a high resolution, the other key feature that really allows us to do to investigate the cultures all the way to the single cell level is the extremely low noise readout. So we just have 2.4 microvolts RMS of readout noise. And this really allows us to track not only the signal at the soma, but all the way across the axon. And last but not least, just in case you didn't have enough, each electrode can also be used for stimulation, up to 32 at a time. So what are we going to see today? I would say these are the three main assays that we're providing and we're going to see in detail today. The first one, the activity scan assay, works as follows. So what you have here at the top left is a simple fluorescence microscope image of neurons growing on top of the chip. And what you're seeing at the bottom is what we call an activity scan. So basically, this figure is color-coded from, I would say, dark blue all the way to red in terms of the cell's firing rate. And basically, with this first assay, we're able to identify where the cells are on the chip, but in a completely label-free way, and already identify where the most active cells are. At the second level of investigation, so zooming in a bit more into your culture, we have the network assay, in which basically we, let's say, we use the readout of the first assay to understand where the cells are and investigate how they interconnect, so how they are connected with each other. And at the third level, the action tracking assay, we still use the information from the first assay to go all the way to the cellular and subcellular level and investigate basically subcellular compartments, really the velocity of propagation, for instance, of an action potential along the axon. Great, so there's a lot on the plate, so let me stop sharing my slides and start sharing.
Yes. Giulio, maybe while you are loading the software, I would just want to show everyone. So this is the MaxOne system. We are not demoing it today, but just to show you, it's very small. It's like the size of my phone. And this is the MaxOne chip, which is right here. And then I would also like to just start off with some questions in a poll to figure out if you guys are actually working already with MEAs or not yet. So let me just put this up and please answer this first question. Thank you. So this allows us to understand whether you're familiar already with the technology or is this something that is new to you? All right. Thank you so much for answering the poll. Okay. All right. So I'll end this. And we have one more question. I'll just add it here. In case you're interested to either upgrade your MEA or to buy a new system, we're just curious about that. So please also answer here. All right. Awesome. Thank you so much. Okay, I know you're all excited to see the results, so I'll end the poll and yep. Now, Julia, the floor is yours again. Okay.
Great, thanks, Marie.
So just close the poll if in case it did not disappear. Yep.
Oh, no, it's all good.
Okay.
Great. Okay, so we're jumping right into the software. So again, I don't see you guys, but of course, like my team is collecting the questions, so feel free to post them in the chat and I'll answer them throughout the presentation. So to begin with, what are we seeing here? So I already opened our GUI, so basically the scope, which is, let's say, how we are interacting with our system, our well plates. First of all, I'll just ask you to, let's say, focus your attention on this icon here at the bottom right, and simultaneously maybe on my system here on at my left. So this icon is basically the control of the MaxTwo hub through the software. So as you can see, basically this light blue square is the let's say the cover of the MaxTwo system, and through this control, I can basically prompt anything to my system here. So for instance, I can open the cover, as you're seeing here. And I can also release the plate that's being connected right now. So basically this is, let's say, the initial situation. I would load the plate and lock it in position and close the cover again. So right now, basically from here, I have full control. I can check, let's say, on the temperature that I set for the system. I can check on the firmware installed, and that, let's say, everything is working correctly in terms of the system. Of course, you cannot see it from here, but I also connected to CO2, so our cells are ready to go and in the ideal environment to run the experiments. Right now, let's say, let's focus on the icon below. So this one showing our well plate. I already initialized it, but basically what you can do with this well setup is basically to input information that, let's say, pertain to the well plate. So every time I record information and I initialize my well plate, I can, let's say, store information regarding, for instance, the number of cells that we're using. For instance, in this case, as Marie said, we're using FCDI gluten neurons, 100,000 neurons and 20,000 astrocytes in BrainFist. And this is actually very helpful because at the end of my experiment, I can use this information and pull this information to analyze my data. Great. So let's say once this is done, what are we seeing here? So basically at the top left of the screen right now, you can see 3 different views that, let's say, you can use to investigate your cells. So the first one you're seeing, the electrodes view, is really the 2D representation of the chip. So it's a 2x4 millimeter square area, and every, let's say, blue line that you're seeing here in the background is actually let's say, the trace being streamed from one of the connected electrodes. So we have 26,400 electrodes, but we work with 1,024 at the time. And every time you see one of these red triangles appearing, it means that we are detecting an event. The second view, and Marie, sorry, I see people raising their hands. Maybe we'll get the questions in the end. Is it okay?
Yes. Okay. Or maybe we have a pause somewhere in the middle, and then we can start answering some questions, raising hands.
Absolutely. You
Yes.
you tell me.
Okay. Thanks. Maybe you can go ahead until before the assays.
Sounds good.
Yeah. Okay. Thanks.
So the second view, I would say maybe it's a more standard view for, let's say, NEA users. So we have a 6x6 grid of windows, each window containing 29 traces streaming from 29 different connected electrodes. And in this view, you can really go and check each single trace. Actually, if I would zoom in, I could check each single trace and really go and check on your signals in let's say, filtered or unfiltered manner. So we also have a third view, but I will reserve this for, let's say, a later stage. And maybe since we already have someone asking to interact, I'll get the first questions now.
All right. So Xue Jiang, you raised your hand, so maybe we can allow you to talk. I will click the button here, and you're welcome to start speaking. You can unmute, and yep, you can ask us anything. Hello. Just wondering if you are there, or maybe you can write something in the chat. All right. Okay, let's go to our next one. Chris. Hi, Chris. I'm giving you the chance to talk. Ask you to unmute, and yeah, you're welcome to ask us anything.
Hi, this is Chris. Do you hear me?
Yeah.
Yes, very well.
Embarrassing, but I accidentally clicked on the raise my hand. But So it was an
Hello,
accidental click,
anyway.
but at least we have a check now that this works. So that,
Absolutely.
Awesome.
that
Thank
maybe
you
the
for
one who
troubleshooting.
actually raised the hand can speak.
It might be it was the same
Yes.
issue. Don't
Yeah.
worry about
Anyway,
it.
waving to you guys. Hello. Yeah. So Julia, then please go ahead.
Yep. Okay. So I'll just go ahead and just write in the chat if you have a question. Maybe it's easier than the raise and not raise hand.
All
Great.
right.
So now we have let's say seen the first two views. I will now go, let's say, to the actual experiments. So the two bottom tabs you're seeing here on the left. So if I click on the assay tab here, I'm actually prompted basically to my experiments. And what I can do now is clicking on this assay gallery icon, or let's say button here on the top left, I can start running our assays. So starting, for instance, as we would do, with the activity scan assay. So with the activity scan assay, you have to imagine it as a way of going through all or part of the electrodes of each well, all the wells in parallel, and scan them for activity. So it's like microscopy. So the let's say the higher the resolution, the longer the time. So this is exactly the same for us. And you can very easily set this up. So first of all, you will need to decide on the configurations to use. So a sparse 4 will just use 4 configurations to cover all the area. A sparse 7, very obviously, again, will use 7 configurations. The checkerboard, 14, all the way to full, which basically will use 29 configurations to scan the entirety of the array area. Then you just need to decide the time per configurations that you want to use and click on Run Assay. So right now, just for the sake of, let's say, our time constraints, I will just click on Sparse 4 and click on Run Assay. Very important, you need to select the wells you want to run this assay on. You can either select all the wells or just select each single well you want to run this on. And then simply click on Run Assay. So it's very, very, very straightforward. And basically what this would do, if we go back to the electrodes view, is to start with the, well, in this case, the leftmost configuration and scan through the array. So now it's a very quick scan. It will just take roughly 2 minutes. And you can already see now we focused all the electrodes we had spread out just on one side. You can already see that, let's say, there is activity, let's say, spreading out. So these red triangles that you're seeing. And while this is running, I'll just show you, let's say, the difference between, let's say, a lower resolution scan and a higher resolution scan. So this is actually a scan I ran this morning for the first demo. Oh yeah, sorry, maybe this one. Yeah, this one. Sorry, I need, yes, exactly. So this one was a 7x. You can see not all the electrodes were selected, whilst again, what another assay that ran this morning in full scan, you see that how much, let's say, this changes. So here we can really see all the cells in the well. As soon as our assay is done, we can also compare this with a 4x. So it will just take, I would say, roughly 2 minutes, but maybe we can, let's say, come back to this after this is finished. Once this is done, basically you have information on the firing rate, the spike amplitude, again, per well, the active area, the percentage of active area, and all the distributions, let's say, for all the wells or for each single well. And what I will show you now while this is running is that you can very easily analyze this data, simply clicking either on, let's say, the shortcut, so going directly to the analysis, or on the analysis tab. So the, let's say, the last tab we still did not explore. So I'll just click here. And well, this I unfortunately already ran this morning, but let's run it again. So for the same of sake of time constraint, I will just run it on one well. The parameters you are prompted with right now for this assay are very, very simple. So they're just thresholds in terms of firing rate, amplitude, and interspike interval. So you can decide, let's say, from where you want to start considering the data, basically. Then we can just click on Start Analysis. And in roughly 10-15 seconds, this will be completed. And there we go. So this is exactly what you're seeing here. So a first, let's say, quantification for the mean firing rate, the spike amplitude, and the percentage of active electrodes, and the distribution for the firing rate, spike amplitude, and the interspike interval here on the right. On top of this, you also have, let's say, rightly available the statistics for the well in terms of the same quantifications. So from here, you straight away have, let's say, what we have seen here saved in PNG, just in case, let's say, you have a quick presentation to prepare. But of course, you can also export your metrics in either Excel or CSV, both the summary or the full metrics. Great. So at this point, what we would do is basically use the information that we collected at this level to, let's say, further our investigation of the cultures. So again, I would go to the assay gallery and click on the second assay, the network assay.
Giulio,
sure.
Before we proceed, we have a question from David. Hi, David. I'll just allow you to talk, and then you can ask us anything.
Sure.
Hi, thank you very much. So I have two brief questions, actually. So one, it's I have my data. So the configuration files, I have been doing the full scans to evaluate the maturation of the circuit, every week. But for time purposes, because every time I do it takes like 20 minutes. So I did it like the one that covers like 98%, but takes I don't know if it's checkerboard maybe.
Checkerboard.
But the configuration file was very different from the one that I was getting. And when I ran the network, the data looked slightly different. So I decided to go with the full scan. So I guess my question is, what would be the reason to use not full? Is this only just the time? Because the quality of the data, or like how comprehensive is your understanding of the circuit, is better with the full scan, right?
So I would say it's always a trade-off, right? Because again, you're basically emphasizing the importance of high resolution for your data, right? Because in a full scan, you are literally recording from every electrode and therefore really from every cell. At a lower resolution, there are, let's say, for sure, some cells that you're not picking up in between. And let's say, given the activity of the cells, you might have shifts in the, let's say, in the extracted parameters. So I would say the important thing for your experiments, since you will always have this trade-off between the time needed to, let's say, scan the culture and the resolution you will have, is to be consistent throughout your experiment. So if you're working with 7x, you're biasing your result on this, which is, it's intrinsically true. So you should always keep the 7x throughout all the experiments. For instance, in the recent paper, Ronchi and all, 2021, I think they were working with 7x and just throughout the full.
I see.
Let's say the full. But for instance, Bandavaravi and all from Tafti Lab were working with full scans. Just they kept it constant, and I would say that's crucial aspect.
And the second question is more like, I mean, I don't know if it's part of this, but in the analysis, when you exported the data after you run the analysis in the network, the heat maps, depending on the well, they have like different color coded. So is there any way to lock the values in the heat map scale. So because the other day I did some treatments on the wells, so I would like to see it like right away, but it was auto scaling for each heat map. So it's beautiful pictures, but I cannot see any differences. So
clear. So
is there any
right
way to do
now,
that?
yeah, so right now this is still not integrated in the software. So let's say to have consistent heat maps for, let's say, throughout all your experiments, unfortunately not right now, they are self-scaled, like in the assay. So basically what you're seeing, sorry, let me just jump to.
Yeah, I think it's just right now it's the auto scale per run and well. Like there is no way to lock the limits of the heat map for the different wells.
Yeah, so go ahead, Mary.
Yes, so David, not yet, but this is something that we are currently working on. But actually, for the network activity, there is more flexibility in setting, for example, the X and the Y axis. Maybe we can also, train you on these parts separately. Yes. Thank you.
Thank you.
Thanks a lot.
Yes, so let me resume. So we were setting up a network assay. So David, for this one, I would say with the updated analysis, I will also show what Marie was mentioning. So in terms of, let's say, setting the axis for the activity scan, let's discuss. Absolutely, that's a very good point. Yes, so for the network assay, again, it's very straightforward. And it's all based on the results of the activity scan. So the activity scan is our key. So I simply need to click on use activity scan here. And let me use a full scan, again, previously ran, just to show you the difference. This is the result of a 4x, this is a result of a 7x, and this is a result of a full scan. So I think this is also interesting to see. Once I decided on the activity scan assay that I want to use, I just click on show selection options, and I'm prompted with different methods for selection. I would say, especially if you're using the full scan, the most straightforward, so what we're going to use today, is the feature maximization. So the feature maximization is, again, very easy. It just asks you to, let's say, set a selection preference, either on the firing rate or on the spike amplitude. So right now, for instance, we're showing it at the firing rate. I'll select the firing rate, and then I click on evaluate selection. And you see the software already found the electrodes that best superimpose my activity scan. So in orange back there. So you see we selected 1,020 electrodes directly below the neurons having the highest firing rate. And I just need to click on finish. For the sake of the demonstration, again, let's just record for 60 seconds. Usually I would go for 300, but just for today. And again, very simply, you have to remember to, I always forget this, but you have to remember to select the wells you want to record on and click on run assay. So whilst this is running, is basically preparing all the inputs, meaning all the electrodes that I want to connect for all the wells. And of course, I mean, this takes longer or shorter, also depending on the configuration, but let's say in a couple of seconds, this will be set automatically for all the wells. And once this is done, we will move, basically, you see from this last configurations that we had to the network configuration selected. So maybe, Maria, I know you have some polls, so maybe while this is loading, we can go for those.
I do have some more questions, but maybe before that, for those who joined us later, I'll just show a slide. I'll share screen, and oops, Giulio is sharing screen.
Oh, sorry, let me stop sharing.
Okay, so just really briefly, just to show you some events where we will be in. So we have started traveling. So I think Giulio will go to Germany by end of this week. So we are joining the physiology conference in Frankfurt. And we have the TED annual meeting here in Switzerland. And then we have the online meetings by middle and to end of October. And just to let you know, as again, if you would like to contact us directly, or if you have any questions, you can email us in this email address. You can schedule a call with one of our application scientists or Giulio directly. And you can also just check out our website on www.mxwbio.com. So yeah, and as I showed here, we have our 5-year anniversary this month. So continuously until the end of the year 2021, we will have some special offers. So if you would like to purchase a system Max 1 or Max 2 within this year, please contact us because we might be able to give you some offers. Okay, so I'll stop sharing this. And then while waiting, maybe we can also answer some questions already asked now. First, okay, so we have another question by David. So here he asked, can you use previously recorded activity scans and network analysis to do the axon tracking assay? And maybe this is something that you can answer later on when you describe the axon assay. Okay, so moving forward to other questions. Let me just go here to our list. Hello, Isam. Thank you for the question. So, Giulio, you mentioned about our 24 well plate format. Is this officially a product? And maybe you can just mention how it is, like more information about the 24 well plate to our audience right now.
Sure, absolutely. So it is officially a product, yes. And I would say, as I mentioned before, it features exactly the same chip as Max 1 and Max 2, 6-well plates. Right now, let's say the 24-well plates will be available by the end of the year for people that already purchased it, and early next year to the general public. Let's say one key information I would say is that we're reading line by line or row by row, so 6 wells at a time. But you will use exactly the same system. So your good old Max2 for the 6-well plates will also be able to read the 24-well plates.
Thank you so much. Yes. How is the network going?
It's already finished, actually.
Okay,
I'm sorry.
okay. So then we can go ahead.
Yes. Yes. So maybe I can also just repeat the essay but let's say maybe for the, yes. So maybe I just wanted to focus very rapidly on this one. So you see basically we loaded the configuration in each single well, and you see we moved from this last configuration we had here on the right to the configurations you saw when we were setting up the assay. And basically, again, it was just a very rapid recording, but already in the summary, you see basically, well, actually, sorry, let me first go to the view so that you know what we're seeing. So basically, this view, I would say, is crucial for our network assay. It's let's say our raster view. What do we have here? So on the y-axis, we have all the connected electrodes, and on the x-axis, we have time. So of course, these cells are really great because you see they're extremely, let's say, irregular. You can see them bursting very rapidly. So basically, whenever you see a dot appearing here on, let's say, on the raster plot, it means that we are detecting an event. And when you see this accumulation of dots is a burst, just for, let's say, people maybe not familiar with this. It means that all the cells are being active together. And you see, basically, these cells are extremely regular. You can tell this, of course, visually by the raster, but also from, let's say, the network activity profile that you're seeing here at the top, which basically is the profile of the bursting activity. So what we are seeing then at the end of the assay we just ran is exactly this. So per each well, here at the top, the raster plot, and here at the bottom, the network activity plot. And as you can see, it's extremely reproducible across all the wells. And again, what we are, let's say, what we can do is jump directly to the analysis. And again, just for the sake of time, analyze only one one well. So the parameters you're seeing here are a bit more complicated. So I'm happy to elaborate more on these if you, let's say, have any questions. You also have, let's say, visualization options that allow you to, let's say, tailor the plots that you're outputting. And this goes back to the question we had before. But again, this culture is very regular, very beautifully bursting. So I am quite confident that if we just start the analysis, we will have, I would say, straight away a good, let's say, interpretation of the result with the standard parameters. And that is correct. So what we're seeing here again is the raster plot, the network activity plot, and for histograms showing you the number of spikes per burst, the burst duration, the burst peak firing rate, and the interburst interval. And again, here you have not only these visualization, but also the statistics per well. You have, as always, the quick PNGs for your presentations. And when you click on export metrics, you can export this directly in Excel and CSV and or CSV and feed it directly to your own statistical tool. Then I would say we can go one step further in, let's say, within our culture and check on the single well level. So before going to the assay, I would like to show you, let's say, with the high resolution. So for instance, if we zoom into, let's say, this area, for instance, this morning I took another one. So let's see what this reserves. And we generate, let's say, a high resolution plot within this area. And I simply click on route. I will basically now just ask the software to connect the electrodes within this area. So you will see them turn into light green as soon as we have it. And if I click on download, basically you see we have the electrodes in this area being now connected. And maybe I'll just, with this high resolution, it's actually quite nice to show it in a visualization. Maybe it's also, let's say, more standard for many users. So this basically I'm just representing now the minimum spike amplitude detected by the electrodes. I'll just superimpose the traces so we know where they are. And basically this is, I would say, the basis of the assay we're going to see now. So you see with this high resolution, we are now seeing the single cells firing. The reason why I can say these are single cells is because I can see the activity being, let's say, collected by many neighboring electrodes. And if I actually click on this footprint tab here on the right and click on add, I will, oops, sorry, I didn't actually click. Yes, click on add and maybe zoom in a little bit more. Yes, and also increase the scale. What you see forming in the back is basically the footprint of, let's say, the functional footprint of one of these cells. So basically signals being collected by this electrode of the signal coming from, let's say, the cells that we're seeing right now spiking. But of course, at this level, we are still not spike sorting the cells. So we're basically just, I will not go into the details, but basically every time there is an event in this electrode, I'm collecting all the signals from all the connected electrodes. And you see, I can easily see where the signal propagates on the, let me just stop this visualization for a second. I can very easily see where the signal propagates on top of the electrode with this triphasic signal going more toward the axon and this very strong positive signal going more toward the dendrites most of the times. But again, as I was saying right now, these are not spike sorted. So we are not, of course, we cannot claim right away that these are single cells. Let's see the signals we're seeing. So to do this, what we would actually do is jump to the third assay, which is the axon recording. So the axon recording assay is again based on the activity scan, which is again our key. And the way in which we do it is to select, let's say, the 20, 50, 100 most active neurons that we are individuating within the culture, and then using these as, let's say, focal points, and keep them constant when we scan the array. And basically then interpolating the results that we obtain from this readout, we can then go all the way to the single cell level. And again, right now, I'll just jump to the analysis. And again, based on the activity analysis, with this assay, you have 3 steps. The first one is the spike sorting. So at this level, we really, let's say, separate the signals coming from each single cell from each other. And we can extract single footprints from single axons. So for instance, let me scroll down, for instance, neuron 16 here. And again, here you have the results and the statistics per neuron here, and again, in your results folder. But then I would say most impressively, in my opinion, is that you can, with the third step, really zoom in into, let's say, not only the well parameters at the single cell level, so extracting results for the neuron conduction velocity, the total axon length, the longest branch, the longest distance from the initiation site, that is basically the distance from the action instance segment to, let's say, the end of the tracked axon, the longest latency, and the amplitude at the initiation site. But also, and for instance, you will see it if we go to the results folder here, really go to single branches of single neurons and check on the velocity of propagation of the signal on single branches within your neuron, which I would say is quite unique with this technology. Yes, so I mean, I know we are maybe a bit time pressured, so I'm sorry if I rushed this a bit, but I would say from here we can start taking questions, so I'm happy to show more.
Awesome. Thank you so much, Giulio. It's really super exciting to see these results from the axons. And yeah, just imagine for every well, you can actually extract single cells and be able to understand how the processes are and also extract the conduction velocity. So guys, I have my last question. So now you have seen kind of most of the features that you can do using MaxTwo. So my last question is this one. So we are happy to have a demo in your location using your own cells. So would you be interested in getting a tailored presentation or a demo using your own cells by one of our application scientists? So we would be happy to hear about your feedback here. Awesome. Thank you so much. And while this is happening, maybe if some of you have questions, you can write them down in the Q&A section. We will answer them live. If you would like to ask Giulio specific things on how to use the software, you can also do that. Or in applications, any applications in general, you can also ask Giulio. All right. Thank you so much for participating in our poll. And now let's go to the questions. Okay. So we still have a question from earlier. This from Bulent. Hello, Bulent. How is your experience with organoids and spheroids? Can you record reliable measurements at the interface of these 3D organoids onto our 2D electrodes?
Great. So thank you so much for this question. So the short answer is absolutely, but actually I have data, so it's not live, but it's that I can replay coming from organoids. So let me just remove this and load the data. So right now I'm just replaying data we recorded with the system. So I just hope, yes, perfect. So I hope you can see it. I know it's maybe a bit faded, but I would say the system is actually extremely cool when it comes to recording with from organoids and spheroids, because the advantage of this technology is that wherever you place the organoid / spheroid, you will be able to record from it because you can strategically place the electrodes just below it. And this is actually what we did here. So let me try to zoom in a little bit. So this data is actually present in our organoids brochure. So let's say we will also send this out after the presentation. But basically what we did here was place 3 different organoids on the chip, and you see basically very nicely how let's say we're recording from them. And I would say not only the density of electrodes is an advantage for organoids in general acute applications for this system, or acute, let's say 3D applications with this system, but also the signal to noise ratio. So this is actually, in my opinion, even more important if possible, because thanks to the signal to noise ratio, we are able to record up to 100 micrometers within a 3D structure. And and very precisely from single cells. And this allows, for instance, to bypass the layer of dead cells, for instance, in brain slices, but in organoids to really go in and record from single cells. So I would say maybe not from, let's see, we're tracking the full extension of the axons can be difficult because of course it depends where the axon goes, but in terms of patterns of single cells is absolutely possible. And again, I will just, well, maybe we answered the other question. I will also again add some of these single cells to show you exactly this. So let's say long story short, absolutely, we have very good experience with organoids and happy to share more results also in a separate call.
Yes, and if you would be interested to have a demo using your own organoids, just please contact us directly by email in this email address info@mxwbio.com, and we would be happy to arrange this demo with you.
Absolutely.
Okay, so yes, thank you so much, Bulent. Now we have another question here. So David, I will go to you after this another question. So hi Liz. I love the high resolution box feature. The accent tracking analysis also is intriguing. Did I understand you correctly that you can track axonal branching morphology via the electrical activity? Can you also track the dendritic arbor? So these are the 2 questions.
Okay. Yes. So yes, you understood correctly. So you can track basically single axons with the system. And again, this is thanks to the high resolution. So basically, when you spike sort, you're able to understand where the signal is propagating on each single electrode. And maybe we can go back to the
results, or maybe the results of the assay.
Absolutely. So let me just go to the results. And just one branch, for instance. Yes, so you see here, this is actually what we're doing here. So this is the morphology of one single neuron. And, if we scroll through, this is actually what you're seeing. So for instance, this one I highlighted in red is basically the signal being tracked on all the electrodes starting from this point to basically the end point. So based on this, we are then able to, first of all, extract the functional morphology. And I would say this is an extremely cool feature, especially if you're working, for instance, with compounds that affect the functional morphology of your cells. So for your cells, for instance, there are publications with hiPSC-derived dopamine neurons that use compounds that induce hypertrophy within 3 days. So with this technology, for instance, you can pinpoint a subset of neurons and really track how the morphology changes throughout the development, which is extremely unique. On top of that, of course, you can also do, as we're showing here, detection of the velocity of propagation. So this, can be affected or can vary based on the age of the culture. So usually in development, the velocity increases and then plateaus. Especially for iPSCs, usually gets into the range of more mature neurons, like primary neurons. But also, it can be affected by, let's say, diseases, or for instance, diseases you're modeling. For instance, Parkinson's, Alzheimer's disease could have some of these effects on the axons. For what concerns, sorry, I'm going a bit long, but I really like the subject.
All of us, yes.
For what concerns, let's say, detecting signal from the dendrites, usually with this technology, you see, due basically to the balancing of charges, you see a strong negative signal close to the soma, a triphasic signal on the axon, basically due to the propagating signal, and a very positive signal in correspondence of the dendrites. And this is actually quite nice. There's a paper out in which this was shown in Purkinje cells in cerebellar slices, and you can really see the fan of dendrites protruding from the soma with these very positive signals, and the soma with very negative signals. So you can even see the orientation of the cell in the slice. Hope it answers.
Yes, thank you so much, Giulio. Yep, and now we'll welcome David back to speak. Hello, David.
Hi, thank you. So I have many questions, but I don't want to hijack the session, and I keep it brief. So I think Giulio kind of answered my question that I had in terms of these high resolution recordings that axon tracking. I think it's kind of hinders by the fact of having other cells, say astrocytes or three-dimensional culture. So it's better in like flat 2D primary, say, cultures. Or you can also do it in iPSCs with astrocyte in there.
Yeah, so sorry, because I hear you faintly. So just let me repeat your question just to be sure I got it. So you're asking whether this is possible only in monocultures or whether this is possible, let's say, also in co-cultures?
Yeah, exactly. As I understood that you can record up to 100 micrometers from the electrodes, but I guess that also affects the resolution for these high resolution measurements that, velocity of conduction, say.
Okay. So sorry, I didn't mean to cut you off. Go ahead.
No. That's the, yeah, you get it right. Yes.
Okay. So actually, no. So it doesn't really affect the readout. And actually, these cultures we're seeing here are all co-cultured with astrocytes, for instance.
Oh, great.
What we're seeing is that actually the presence of astrocytes really, let's say, helps our cultures to grow nicely. So usually we see the onset of activity a bit earlier on in the presence of astrocytes. We also tend to see, let's say, an effect maybe on the amplitude of the signal, meaning we see larger amplitudes in the presence of astrocytes. So I would say it's the contrary. So if you have supporting cells, it's actually much better for your culture. So I would strongly suggest having that.
And then the other two questions is just basically if you will comment on the assay that's called record. So just do recording. So how different is from network analysis if you load a configuration file in recording? Yeah. And if you can combine the stimulation with the axon tracking. Those were my last two questions.
Okay, so two questions. The first one is, I would say, easy. So the record assay. The second question is if you can combine the axon tracking with stimulation, correct?
Yes.
Great. Okay. So no worries. It's a pleasure. So the record assay is very straightforward, and in my opinion is an underestimated but a very cool assay. So the record assay basically works like this. So you can load, let's say, the configurations that you are interested in. So let's say you have a network configuration and you want to record over the weekend. Let's put it like this. I don't like working during the weekend, so I want the software to work for me. So what I do is just load let's say, the network configuration, for instance, the one we had before. Let's say, I would say here in well 2 will still be there. Oops, sorry. Yeah. So for instance, I want to load this configuration, and I can do this basically either loading the one that is already present or going through the configurations files. And then I can set the recording time per recording session. So let's say like this 5 minutes, the time to wait in between recording cycles. So let's say I want to record every 2 hours. So here I would just put 120 minutes, and then the number of recording cycles. So let's say I want to go throughout the weekend. So I'm going to put 24 here, let's say, roughly. And then I just click on Run Assay. So this basically allows me to have the time points I want automatically for the system. Of course, you know, maybe this defies a bit the concept of high throughput. So, you know, if you're not there, this just works for one well plate, the one that is inserted there. But this really allows to run basically the same configuration over multiple times. Could be useful also for high resolution recording, just in case you want to always track the same cells, for instance. I don't know, Marie, if you want to add something on this assay.
All good. So basically, it's similar to the network recording, or typically how you would use a network recording. Is just something that you can schedule longer. Yep.
Yep.
Yeah. And the last question was the stimulation.
Yes, exactly. So the last question, it's I would say opens many doors. For instance, one thing that is you know, for instance, possible to do is having individuated single cells to combine basically this outcome with stimulation. So you could potentially let's say find the footprint of your cell, individuate which electrodes are involved in this, and for instance, stimulate at the soma and read on the axon, or vice versa. But this is still not integrated in an assay, so it opens more to, let's say, exploratory approaches, or let's say something that I would say was very common, for instance, the lab this originates from on MaxOne, maybe less common on MaxTwo, but still possible.
Mm-hmm.
I hope it answers your question.
So currently the axon assay depends on the spontaneous activity of the cells. So it's not yet something that is evoked by stimulation, but this is something coming in the future. So yeah. Okay.
Yes, sorry, I hope you misunderstand the question. That was also one way of answering that.
Yes, yes. Thank you so much, David. And then I'm just checking, do we have other questions from our audience? Let me just check if we missed anything. I think we have one question that came in by the registration, and this was, is it compatible with microfluidic chips?
Right. So let's say the short answer is again yes, but it requires some integration. So let's say based on let's say your chip, for instance, or what you want to integrate, we might, let's say, need to really interact. This is something we actively do to figure out how to do it. But otherwise, yes, it has been done already. It is possible. Of course, it requires some tweaking, maybe on both sides.
Yes, because also the size of the array is quite small, so you would probably need a special clean room or just a method on how to fit that. All right. So oh, we have one question here. So this is another question that we got from the registration, comparing your in vitro results, because now we have human iPSC-derived neurons. So comparing these results to real life human testing, can you make such a comparison?
Right.
Well, he asked here with a smile. The question is asked with a smile, yes. Thank you so much for the question.
I would say this is, you know, maybe less on the technology, more on what we're working with mostly, which are iPSC-derived neurons. So let's say patient-derived neurons really, let's say, exhibit activity that can be, let's say, related to the, let's say, in vivo situation. I'm thinking, for instance, I don't know, retigabine, trials, but also all the studies in pain, for instance, with nociceptors. Let's say you have let's say you are able to create a model that can be translated to the in vivo situation. So that for sure is possible. A seizure, for instance, very similarly. Of course, it's not a one-to-one. It's always a model. So I would say it's up to the researchers to interpret the results and let's say find their way.
Yeah, so of course, not for behavioral studies because we cannot see the behavior in vitro. However, if you want to test compounds or if you want to check if there is an effect of the drug for the specific disease so the cells coming from the patients, I think this is something that is getting more and more possible thanks to the iPSC technology. But still, we have a lot to look forward to in this technology, also in this field for making it much better translatable for human real human use. Yep. All right. So I guess that's all of the questions. Thank you so much, everyone. Thank you for participating. It was a very lively event that we had today. Super nice. Really appreciate it. And again, if you have any questions, please reach out to us. You can see our email address here in the chat, and also just contact us. Also, Giulio here would be very happy to have a demo with you. And again, we would be very happy to talk to you personally for your own applications. So please reach out to us. All right. That's it. Thank you so much for joining our webinar, and see you again soon. Bye. Thank you so much.
Bye-bye.
Thank you.